Hi guys, I'm gonna do a dry run of my presentation here, just so you guys can comment on it. It's a presentation about my research background. So I started my career in neuroscience on the Comparative Neuroanatomy Laboratory. It was a laboratory that studied the brains of various mammals, characterizing them on the cellular level, using a newly developed technique called the isotropic fractionator that would pick up these brains, dissolve them in detergent, and assuming that cells have one nucleus and one nucleus only, you would have a suspension of nucleus after you dissociate them in a tissue grinder. So if you simply agitate those and mark the neuron nucleus with antigen like neon, for instance, you can count them on a microscope and see how many neurons a brain has. So in the laboratory we quantified a very odd phenomenon, which is the loss of neurons after birth in mice. If you look at these graphs that I'm showing you, the x-axis is days after birth and the y-axis is the density of neurons in various regions of the brain. So you can see that in all these regions you have a significant loss that accounts for more or less 70% of the neurons in the structures. The same happens if you look at synapses. This is a quantification of dendritic spines over time in primates, and you see that even though they reach a peak around 3.5 months, they decrease a lot over time. And even though this was extensively characterized in the cellular and molecular level, we still don't know the effect that this loss has on the network, why evolution would shape the mechanism in this way, in a way that it would waste or use a lot of energy to begin with, and then you just cut and kill cells. So how do you study networks? We can borrow a framework from graph theory that turns the neural network into a series of nodes and edges. Nodes representing the cells, representing the neurons, the edges, the interactions or links representing the synapses, and you can then count how many connections each of these neurons has, and we call this degree. So you can have an in degree, which are the connections coming to that cell, and an out degree, which are the connections going out of that cell. So how would you expect a network like a brain network or a cortical column to be? Well, if it was a random network, we would have a degree distribution, a distribution of degrees over nodes in a Poisson way. If it was a scale-free network, we would have a Poisson law. What are the advantages of a Poisson law, and why would we think that a scale-free would be more likely to be the network that the brain would present itself with? Because it has a high robustness, which means it's really resistant to attacks. If I delete nodes, if I kill cells, if I have a stroke, or if I lose a limb, those regions can adapt and change function. So I will have a short average path length, which means I can move from one place of the network to other place of the network really quick without having to jump in through all the neurons in the network. And I will have a high clustering, which is a high level of modularity, a tight connected subcommunities inside of the network. So to test in a network way how the brain would be if those procedures happen in a way that we think would be plausible, like by selection of the best connected neurons and to keep them alive and to keep the synapses, we came up with this model where we generate a network of neurons and we distribute connections them randomly, and we run a neuronal death stage where the less connected neurons are just ripped off the network using the formula you can see over there for DN. After that, we do a same rationale step, but now for the synapses. So we also cut off synapses according to how well connected or not that neuron is. The only difference between steps 2 and 3 is that on step 2 we actually reintroduce those cells and connections in the network, so we would keep the size of the network stable. On the pruning stage, no, we just got connections that are not from the most connected cells. And we run 1,000 iterations, even though with 500 more or less the network gets to be stable. So what happens when you have preferential depth for the depth stage and preferential pruning for the synaptic stage? When you kill cells in the preferential fashion, you end up with a fat tail like the Poor law that we saw a couple slides ago, but not the less connected nodes. They don't follow the Poor law fashion. After the pruning stage, we have something that resembles really a Poor law. So neuronal depth, if it is preferential, and synaptic pruning, if it is preferential, generates a degree distribution that's very similar to a scale-free network, which is what we would expect for the brain. Now, what is driving that then? Is it the depth? Is it the preferential depth that's driving that change into a Poor law? So let's try do a random depth, and you see that when you do a random depth, it changes very much the degree distribution from the preferential depth. And after that, if we run synaptic pruning in the actual preferential fashion, we go back to the Poor law stage. So, the depth doesn't really play a role in shaping that Poor law. So, could it be that if we remove the depth completely, would it still be a Poor law? So that's what we done next. We removed everything. Again, I have the distribution from the beginning, just after one iteration. And with the synaptic pruning, we go back to a Poor law. So the depth plays no role whatsoever. So what about the pruning? If you run random pruning with 3 random interactions, the network is sparse. After 600, the connections are almost gone. So the pruning is key, but the depth is not. So last variable you have to test is the feed-forward fashion of the network, because we described the network in the beginning using a feed-forward fashion, which means the nodes are connected in order. They are always connected in the same direction, from one to another and not on the other side. So the removal of the feed-forward of the network, if you have no feed-forward, you lose, even with preferential attachment, preferential depth and synaptic pruning, you lose the Poor law distribution. When you go to, like, again 80% of feed-forward again, you go back to almost what the Poor law would be. So this was the project I was involved in, that was with computational modeling. Now I'm going to talk about something different. It's my vision for this project that we are going to develop here. So the project is about modeling S1, the somatosensory cortex, and testing different learning rules to see how plasticity would appear in a way that we could test with actual experimental fMRI. So the way I see it, using my background in network science, it is a modular network of simulated neurons. You have here on this image different subsections of the region 3B, because I chose to assume that the hand would be the input, like the feedback from the fingers, the fibers from the fingers, so I chose to simulate 3B only, from D1 to D5. So if I have populations of simulated neurons for each one of these communities here, I can extract population data, LFPs, in a way, and then with the HRF function, with the blood flow function from a balloon model, for instance, I could convolute the both of them and get to expected boat signal. And that boat signal I could actually test against the boat signal from experimental fMRI. Of course, it depends on the protocol of the fMRI and what you're looking for, but we can simulate it. And after we apply different inputs, different sorts of learning rules and inputs to this network, we can see how these communities change, how they change in size. And this change in size and importance of these nodes can be seen on a baud signal. Too, you can see the change and the variation in the sizes of the regions on an fMRI, on a baud signal. So that's what I think about the project. Thanks for watching. Leave your like. No, that's not a YouTube video. And yes, I forgot about the feed-forward thing in the beginning. Lastly, I will talk to them about my skills, the summary of my skills to the group. So the languages I know, Python, C++, Lua, MATLAB, and especially my network science background, and my knowledge in neuroanatomy and experimental electrophysiology that helps me on the simulation to do a biologically driven model of it, apart from the coding and the coding of neural signals that will be built into the generation of the LFPs. Thanks. This is supplementary material.